Gisela and I are here talking for a real team. You'll understand when we start talking that there's one heck of a lot of work, and it's not two people who have done this. Various postdocs and graduate students of various places, very international. These are graduate students and postdocs who've been in our lab, as well as people from the Charles Darwin Research Station. So we're going to the Galapagos tonight. And where are the Galapagos? They sit here off the coast of Ecuador. They belong to Ecuador. They're about 1,000 kilometers or 600 and some miles off the coast of Ecuador. And what we're going to be talking about are these giant tortoises that live there. The Galapagos giant tortoises are one of two lineages of giant tortoises that have survived today. They only survive on oceanic islands in the Galapagos and Aldabra. At one time, giant tortoises roamed every continent except for Antarctica. Almost all of them went extinct by the Pleistocene, and only these two lineages of giant tortoises remain today. And by the way, they're not particularly closely related to one another. They're not phylogenetically very close to one another. But we're going to be talking about these. These are kind of dull. They're not very differentiated. And we've done a few, or should say Gisela's done a few studies on these, and they're not nearly as interesting as what we'll be talking about. It's really true. Well, they're morphologically and genetically very, very similar from place to place. Anyway, the Galapagos Islands, for those of you who don't know them or haven't been there, sit exactly on the equator. The equator passes through them approximately here. There are 12 big islands. There are many smaller little rocky outposts that come up here. They are volcanic in origin. They've never been connected to the mainland of South America. Many of you may be familiar with the geology of Hawaii. It's somewhat similar in that there's a tectonic plate that's passing somewhere 3.5 million years old, and the youngest ones are here in the west. Fernandina is not more than 500,000 years old. So it's about the same time frame as the Hawaiian archipelago. But as you see, it's not nearly as nice and linear as the Hawaiian. It's a little bit messier here. But the younger ones are here in the east, the older ones in the west. And from these aerial photographs, you can see they really are volcanic in nature. Each is a different volcano, but this big island is in fact 5 different volcanoes that have coalesced to one another, and that's this big island of Isabella here. A couple of differences from Hawaii. Number one, as you can see, it's much drier. Fresh water is at a premium in the Galapagos. It's much, much drier. Second of all, humans did not ever, as far as we can tell, there's no record of any human habitation there or any humans there until the Europeans stumbled upon it in 1535. And so the experience of humans, or the experience of the organisms that live there with humans, is very, very recent. Of course, they're famous because of this chap, Charlie Darwin, who, in the famous voyage of the Beagle, stopped in Galapagos, and it was one of the prime sort of inspirations for him coming upon the idea of evolution by natural selection. And of course, the Galapagos have all of these magnificent organisms. I'm just showing animals here. Sorry for the plant people. Many of them, or most of them, the majority of them are endemic. That is, they exist there and nowhere else in the world. There are various birds and these reptiles. One thing that you'll notice here is a lack of mammals. Except for this sea lion here, which actually goes up and down the coast of California and is very widespread, there are virtually no mammals in the Galapagos. It's reptiles and birds that are the main vertebrate fauna. And this guy up here is actually a Galapagos hawk. It's the only predator in the Galapagos. It's the only native predator in the Galapagos. Those are magnificent animals. But what we're going to talk about, of course, are these tortoises. The genus is Geochelone, and it's been given various I'll say something about taxonomy maybe near the end. It's a mess, the taxonomy. It's given different species names, Nigra, Elephanopus, but Geochelone is the genus. 15 species have been formally described, given Latin names, 11 of which still exist today. 4 have gone extinct within human times, within the last few hundred years. And most of them correspond to a taxon on a single island. The exception was that big island of Isabella, where the 5 volcanoes that have coalesced, each of those has a named taxon on each of the volcano tops. Most of these, because most of the moisture is high up, the tortoises are pretty much confined to higher elevations. Okay. The main morphological variation that impressed Darwin is in this carapace or shell shape. There are 2 major morphology types, one of which is called saddleback, which has a very high opening in front here. Presumably, and these animals usually have longer necks, presumably they can reach higher up. It's sort of like a giraffe kind of story. They can reach higher up on the vegetation with this high opening like this. It's called saddleback because from the side they reminded the Spaniards of Spanish saddles. The dome type is more typical of a tortoise. The frontal opening comes down. These are actually in cooler habitats. Presumably in lusher habitats, they don't need to get as high. Also, the low dome, presumably, the idea is that it conserves heat better than this. So in cooler habitats, these predominate in drier habitats, this type. So this is a main morphological difference among the taxa. The importance of tortoises in Darwin's thinking is sort of illustrated here when in 1959 the British issued a memorial stamp. Of course, 1859 was the publication of the Origin of Species, so this was a centenary of the publication of Origin of Species, and when they did their stamp, they put the tortoises on it, which makes us feel good that the British think the tortoises were pretty important to Darwin. Okay, so what's the problem with their conservation at the present time? Well, various things. For example, buccaneers and whalers, the waters around the Galapagos are very rich in fish and whales at one time. And lots of whalers came there to do whaling, and the buccaneers used to raid the coast of South America and then run out to the Galapagos and hide. And so there were, in the 17th, 18th centuries, a lot of whalers, buccaneers came, and they were eating the tortoises. The tortoises can remain alive aboard ship for 6 months or more with no food or water. So they would go to these islands, collect 50, 100, sometimes 200 of these tortoises, throw them down in the hold, and every so often bring them up and have fresh meat. And from all accounts in the log books, the meat's actually quite delicious. I've never tasted it, but presumably it was quite good. And in days before refrigeration, it was a source of fresh meat on these ships. But more lasting than the fact that they were taking them for food was the fact that they introduced animals. One of the worst types are goats. As you can see, the goats in this island can get extremely dense in population where they compete with the tortoises. Also cats, rats, and here's a case where a tortoise has been chewed on by a feral dog here. So it's the introduction of these foreign animals that never existed there that have been really the lasting impact on the population of tortoises. As I told you, there are virtually no native mammals. So you start bringing in goats, cats, rats, dogs. You got all these burrows and things like that. So those are the problems that are faced today. People don't eat them very much today. I shouldn't say never, but it's the introduced animals that continue to be the problems. Okay, we got into this, as Jack said, in the early 1990s when the Charles Darwin Research Station, along with the Galapagos National Park, the whole area is a national park. The Charles Darwin Research Station is a private organization where all the scientific research is coordinated out of. And they were conducting a census program of the remaining natural populations of tortoises, the 11 species that still exist, and they were out measuring them, weighing them, and these are the park rangers out doing that. And we were tagging along and taking blood samples. And here's taking the blood samples. It's more or less the same way that you would give blood in the doctor's office. We get the vein right about here. You have to pull that arm out, which some of these things are pretty darn strong. It can take a couple people to pull them out, but we get the blood. And remember, reptiles have nucleated red blood cells, so there's plenty of DNA in them. I think we have close to 4,000 blood samples now from both natural populations and some captive animals that we'll talk about a little bit later. So we have the morphology, and they're now putting in these passive internal transmitters to mark them as well. Okay, the first thing I want to talk about is where did these guys come from? Okay, what's the origin of the Galapagos tortoises? Well, on mainland South America, there are three congeneric species, three species of the same genus, Geochelone, on the mainland. And so those were clearly the candidates for being the origin of the tortoises out here. And the currents along the coast here come up the coast and almost exactly at the equator turn out west. So it's been long thought that the tortoises probably rafted or drifted out to the Galapagos by the currents, but which one of these is the closest relative? Well, that was our first little venture into this, and we did a little bit of sequencing, not a lot for those of you who do sequencing. This is all based on DNA sequencing. That's how you do genetics of tortoises. You don't do crosses like you do with fruit flies, but you look at their DNA. And our conclusion from this initial small data set is that Geochelone chilensis is the closest living relative to the Galapagos tortoises. These are all Galapagos populations, from the different islands. And chilensis is clearly, it's also called a Chaco tortoise, is clearly the closest living relative. The other important thing you can see from here is that all of the populations or species of Galapagos tortoises are descended from a single ancestor. That is, they're monophyletic in origin. So all of this happened on the Galapagos by the founding by a single lineage of tortoises. And I'll say a little bit more about maybe the timing of this here. We did a little bit more sequencing, and we included some more populations. And here was one of the more, what should I say, striking or shocking results we had. And that had to do with a population, I shouldn't say a population, an individual of tortoises that live on this kind of remote island of Pinta up here. This island, as you can see, is remote. And it has a single individual left in this species. Okay, here he is. He's commonly known as Lonesome George. I don't know how many of you ever remember George Goebel, but he was Lonesome George. He was the last one found in Pinta. He was brought into captivity. He's at the Charles Darwin Research Station. He's been there for quite a few years now. He's with two lovely females, but he doesn't seem to be interested at all. In fact, he's made the Guinness World Book of Records as the loneliest creature on Earth. So he's the last of his type. And actually, this is from the swimsuit issue of Sports Illustrated about 5 years ago, that he was featured as being a lonely male here. But anyway, one of the important things is that George is sort of the symbol of all conservation efforts in the Galapagos. I mean, he's the guy who's on all the coffee cups and the T-shirts and et cetera. He's the symbol. He's an icon of conservation, as he's been called. And so he's really important. Well, one of the shocking things that we had found from our initial DNA sequencing was that Lonesome George here from Pinta is virtually genetically identical to another population of tortoises from Espanola down here, which is the furthest island from Pinta. So why is his closest relative here, if not close relative, this initial DNA sequencing showed him to be virtually identical to the tortoises from here. And it turns out that actually when we told these data to the Grants, Rosemary and Peter Grant, they said, oh yeah, that makes a lot of sense because the currents, the prevailing currents go more or less in a southeast to northwest direction in the Galapagos, and it's certainly possible that an animal drifted from Espanola up here. Alternatively, it was possible that maybe Lonesome George is not the last of his species up here in Pinta. Maybe he was a recent transplant. And we have evidence that we'll talk about later that some tortoises have been transplanted probably by humans from one island to another. And maybe he's not the last of his species and he's actually just a transplant from Espanola, which if true, would have made us very unpopular in the Galapagos if we had to tell them that their icon of conservation is really not the, we sort of scratched our heads and how would you explain this to people? Well, fortunately, we got around that problem. Oh, and by the way, morphologically, the Espanola and the Lonesome George are extreme saddlebacks, so morphologically, they're very close as well. So you couldn't really tell them apart morphologically either, so they're very similar to one another. Well, the museum came to our rescue. Very, very important that museums did this. This is the California Academy of Sciences. In the early 1900s, the California Academy of Sciences sent a lot of expeditions to Galapagos and worked on Galapagos organisms. And what did it mean to work on organisms in the early 1900s? It means you killed them and stuffed them and brought them back to your museum. And so they had specimens that were collected in 1906 from Pinta, and they were in good enough shape that we were able to get enough DNA out of them to see what they looked like. And the bottom line is these were virtually identical to George, thank goodness. And so we can say that George is the last of his lineage that existed on Pinta at least for 100 years or more. So we didn't have to say that he was not the last of his species, but these museum specimens were important. We did more sequencing. We got quite a bit more data. Just again, technically, it's quite a bit of mitochondrial data. We have some nuclear data, various sorts, including microsatellites. So it's a lot of more genetic data we have. And we have many more population samples. These circles are all the different population samples we have. And I just want to point out, actually, there are 3 samples on the island of Santa Cruz. Again, this is going to play a part of the story a little bit later. But these are all the samples we have, and we've sequenced quite a few individuals. And this is the bottom line phylogeny of what the tortoises are. And what you see is each of these, Pinta and Espanola, while they're the closest relative, in fact, the Pinta, now that we have more specimens and more DNA sequencing, are distinct from Espanola, but they're very close. These are the red ones. These are the oldest populations, and they tend to be to the east, and they're color-coded as to where they came. The big island of Isabella was colonized twice, once from San Salvador and once from Santa Cruz, and each of the named taxa are monophyletic groups. So based on this, we can say that, in fact, these named taxa species, we'll call them for now, are good species genetically as far as we can tell, with the exception of southern Isabella. Southern Isabella here is kind of a genetic mess. We really cannot differentiate what's going on here. Here's the timing. If you believe in molecular clocks, we can try to put a time on these nodes. And the important point is, as far as we can tell, all of the differentiation of the tortoises occurred on extant islands. That is, the oldest islands here are at least 3 million years old, and probably the differentiation of the whole lineage began about 1.5 to 2 million years ago, so that all could have occurred within islands that still exist today. There are other islands that actually are off to the east that have now eroded underneath, but it could have all happened here. And they're all good monophyletic groups, so the taxon, it's rational to think of them as good species. And that's a phylogeny. For those of you who like networks, this is what's called a TCS network. And the color coding, again, to show that each island and each taxon, each of the colors is a different mitochondrial haplotype, are distinct from one another. We like this TCS. The C stands for Crandall, by the way, who may be familiar to you. It's a nice, very nice thing that Keith developed. It also supports our contention. The other interesting thing is that if you look at the older islands here to the east, they have more genetic variation, and most of the variation is among populations. That is, they're differentiated more among populations and relatively little within. The younger islands with presumably the younger populations, much less of it is among populations and more genetic variation within populations and less among populations. So it makes a lot of genetic sense that these are the oldest and these are the youngest, follows the geology, follows the current. The whole thing makes a lot of sense. And I know this is really busy and makes is complicated, but what I want you to notice is the solid arrows all go from the older islands to the younger islands. They go more or less in the same way the currents go. So this is our sort of our way of interpreting the genetic data. These are the oldest populations. We don't know which one of these is oldest. They colonize the next next oldest island. And then, as I say, the big island was colonized twice here, and et cetera. Now I have these little dotted lines here because sometimes in these populations we would find genetic mitochondrial DNA types that were quite homogeneous and quite distinct from one another, except we would find 1 or 2 individuals, sometimes 3 out of a sample of maybe 50, that were completely different. And they would have genotypes typical of another island. And we called these things aliens because they didn't seem to belong to those populations. But what we really think they are cases of human transport recently from one island to another. And we know, again, from log books of whalers and buccaneers that sometimes they did move animals from one island to another. They would collect here, and then they would go somewhere else and see, oh, these tortoises are bigger than those tortoises. Let's dump these and get the bigger ones, or things of that, or leave them here as they went off to the whaling grounds and then come back to pick them up, etc. So we think that there are some human introductions, and as they say, we call these aliens. So that's what we think of the history of this at this point, and I think at this time I am going to pass this off to Gisela, who will talk a little bit more.
I'll be back.
I'm not Jeff, I'm Gisella. And I'm going to give you a few examples of the kind of inferences and insights that we can gather from the analysis of DNA sequences or DNA or mitochondria or microsatellites data. Once that can be gathered only because we have a large database, and we can make inferences that otherwise will not be obtainable if that was not the case. And the first thing I wanted to talk about is an example has to do with the tortoises from Volcano Cedo, a volcano in Isabella, in which the lesson that we are going to learn, that these tortoises are providing us with is that large population sizes does not translate necessarily into high levels of genetic diversity. Usually what happens is that through time, if you have a large population, you're going to have more genetic diversity than in a small population. And in some cases, but what the example I'm going to show you is that that's not necessarily the case. So let me talk a little bit about the genetic diversity of the Galapagos tortoises populations on Isabella. So there are 5 different populations, or taxa in any case, that are on the slopes of the volcanoes. And when we went and looked at the levels of genetic diversity harbored in these populations, we found that the population of volcano Alcedo had a very low, H stands for haplotypic diversity in this case, had very low 0.17 genetic diversity when compared to the other populations. And that was very puzzling to us. Why? Well, for a series of reasons. First of all, let me say that volcano Alcedo is one of the largest extant population in the archipelago. It's between 3,000 to 5,000 tortoises on these slopes. And usually, as I said, the larger population size, the more variability you would expect, or other things being equal. And also, interestingly, it was probably one of the first populations to be colonized. So it's not only large, but it's also an old population, which means that more variability, there was a chance for more variability to accumulate. And also, the other interesting thing is Alcedo being in an isolated situation, it was not exploited by whalers, so that genetic diversity was not exhausted by the fact that the animals were taken away. So all these things considered, it was very puzzling that volcano Alcedo had such a low variation when compared to the other populations. So we went out and tried to explore the reasons behind this pattern. What were the processes that led to this, the evolutionary processes that led to this? So what we did is to compare the genetic diversity on volcano Alcedo with the genetic diversity on 2 other populations on southern Isabella, located on a volcano called Serra Azul and Sierra Negra. And we compared genetic variation in the same number of individuals for the 3 populations, 90 tortoises for each one of them, using a variety of tools. And I'm not going to go into the technical details, but just to show you that this is a network again. These are 3 networks that show the patterns of genetic differentiation for the 3 populations. These circles indicate different haplotypes, different sequences that are present in the population, and the size of the circle indicates how many individuals of that population share the same type of sequence. And you can see that, for instance, in this case, you have many different sequences that are present in the same population. And these steps here, these little empty bubbles, indicate the mutations that separate one sequence from the other. So there is this population, for instance, has 1, 2, 3, 4, 5, 6, 7, 8, 9 haplotypes, and some of them are pretty distinct, separated from by multiple steps from one another. And this is true for this population on Serra Azul, and also the same kind of structure of the genetic diversity is present in this other population on Sierra Negra. But you may notice immediately that the pattern that we see on volcano Alcedo is very different. Here, you have the majority, actually 90% of the individuals share a single haplotype. They have only one sequence in common that's shared by 90% of the individuals. With other haplotypes, they are present in very small frequency and only in few individuals. In this case, it's one individual, here, here, and here. So there is a striking difference between how the genetic diversity is partitioned between for these 2 populations and in relationship to volcano Alcedo. And so what we ask, usually this kind of pattern indicates that the population is very recent. And while this kind of pattern indicates the population is a more stable population that has been, as I said, stable and older population. So this kind of pattern, usually you see them when the population has been subject to what we call a genetic bottleneck, meaning the population size has been drastically reduced by some phenomena, and after that bottleneck, only few individuals have passed through this bottleneck, and therefore the amount of variation harbored by these individuals is much lower than the amount of diversity before the bottleneck. So, but there are tests that you can do to see if they actually there is a signature of the bottleneck in these populations, and we did this test, and we found that while these two populations on southern Isabella, there was no evidence of a genetic bottleneck in their past, the population on Volcano Cedo has had a very high probability of having been drastically reduced in size in a recent past. So there are methods, again, using mitochondrial DNA data and coalescent and statistical methods that have been relatively recently developed to date this bottleneck, to know how long ago this happened. So we did that, and we found that while the two populations southern Isabella, the last common ancestor, basically, of the haplotypes on both in both islands dated back to more than approximately 500,000 years ago, the timing of the shrinking on size of the population on Volcano Cedo was much more recent and was around 88,000 years ago. So not only the two populations, these three populations had different levels of diversity, but these different levels of diversity were related to a different age of the population. So we were curious to know what happened on Volcano Cedo. There was any physical phenomena that could explain this dramatic reduction in population size, this bottleneck on this island, on this volcano. And well, what happened was a humongous cataclysmic eruption that happened on Volcano Cedo around that time, around 100,000 years ago. So we now can tie the pattern of genetic diversity we have observed to a cataclysmic event, which probably wiped up the whole tortoises population on Volcano Cedo. We have records of, like, I think the lava, there was 3 square kilometers of lava that was poured off on the slopes of Volcano Cedo. The tortoises were fried, basically. And what happened is that the volcano was recolonized from a tortoise population from another area, and therefore the recolonization happened with very few animals, and so there was very reduced, and therefore the genetic variation was much lower than the genetic variation on the other two population. And so the take-home message from this kind of studies that modern molecular population approaches using DNA analysis can obtain historical demographic information that cannot be discerned based on contemporary scenarios. Alcedo has 4,000 tortoises. The other two populations, the Sierra Negra and the Cerro Azul, have only 400 tortoises. If I was going just by these numbers, I would have predicted that this population had much more genetic variation than this population. But we know that that's not the case, and we know also why it's not the case, because Alcedo, although it's a much larger population, it's a much more recent one. And we would not have inferred that if we didn't have the DNA data to tell us this. And I just want to go now to another example and talk about the fact that even if you are Galapagos tortoises and you live in the island that is the headquarters of the Charles Darwin Foundation, you can, you can discover hidden variation that probably leads to the fact that probably what we have done here is to discover a new species, certainly a new lineage, and probably a new species of giant Galapagos tortoises. So even if you're famous, it doesn't mean that diversity, all the diversity that's hybrid in this group has been studied and discovered. And this is the case for the tortoises on Santa Cruz, where Jeff he mentioned that briefly. On the island of Santa Cruz, which is also the island that has the largest inhabited city or village, city at this point, and the headquarters of both the Galapagos National Park and the Charles Darwin Foundation, there are three main populations. This is the rough map of Santa Cruz. There is one population called La Caseta, which harbors roughly 2,000 tortoises. There is a small population, isolated population, in a place which is pretty inaccessible. That's the name, Cerro Fatal, because it's really hard to get there. And it's only less than 100 animals. And then there are, I don't even know if you want to call it a population. There are three animals. They are saddleback. Notice that these two populations are domed. They are found on the western side of the island in this location. Namely, there is only one species or one taxon described for Santa Cruz, which is called Caccone porteri. However, there are, as I said, three distinct populations. And so what we did is to look at their DNA and let me say that this is what formally recognized. There are only, as I said, one taxon formally recognized. And while the Central Montura population, it's clearly saddleback, the other two are both domed. So morphologically, they're really indistinguishable. When you look at the patterns of genetic variation and you compare the population from La Caseta and the population from Cerro Fatal, these are, I just want you to look at the brown square. These are the two populations that are domed and morphologically indistinguishable. These are measures of genetic divergence, and both measure the mitochondrial and microsatellite for two different types of mitochondrial DNA data. And the important thing, the take-home message is that La Caseta has much more genetic diversity, 0.80 in this case, than Cerro Fatal, which has 0.08. So much less genetic diversity, both at microsatellites, 15, mean number of alleles is 15 versus 5, and mitochondrial DNA. Also, not only the Cerro Fatal population is much less variable, but also the two populations are very divergent. And this is true both for from this analysis, which is called analysis of the molecular variance, in which it just shows that 91% of the variation is between populations, between La Caseta and Cerro Fatal, rather than within either the individuals in Cerro Fatal or La Caseta. And also by another index, which is called a fixation index, that shows that the levels of divergence between these two both domed populations occurring on the same island, there is a huge divergence. If you put this in a phylogenetic context and you look at the three populations and where they fall on the tree, what you will see is that they are certainly not each other's closest relative. They are not within the same clade. Actually, they are linked to each other by the deepest nodes in the phylogeny. The population from Santa Cruz, from Cerro Fatal, is most closely related to the older populations of San Cristobal, Española, and Pinta, while the population from La Caseta is in a basal position to on a different clade that gave rise probably to the Isabella phylogeny, populations, and the saddleback population from the westernmost part of the island, from Cerro Montura, is very close to the population, which is, again, saddleback from the island of Pinzon. And probably what happened is that either some animals from the population of Pinzon arrived and established themselves on this side, or it could be that this population these individuals are the relics of once more abundant population that colonized Pinzon. We don't know how it went, but clearly they are sister taxa. So based on this, as I said, the two populations that are bloomed, the La Caseta and Cerro Fatal, are as distinct as populations or taxa, named taxa from different islands, but still they are called the same. They belong to the same taxonomic level, either species or subspecies. So what we did is to go from this DNA diagnosis and say, okay, now we know that genes are telling us they're really different things. Can we find some morphological characters that allows us to discriminate between the Cerophata population and the Lacazette population? So we did some morphomatic analysis in which we just measure. We use calipers and tapes to measure different parts of the animals, and we found this work is still ongoing, and we found some differences. In general, the animals from Cerophata are smaller than the one from Lacazette, but it's pretty hard because the differences are very subtle. And what we are doing now is to develop a 3D, a digital system to reconstruct the shape of the carapace. This is a photograph of a carapace of a tortoise, and these are the digital reconstructions. And by doing that, we are now trying to discover differences that otherwise will not be, much subtle differences that otherwise will not be possible to study by using just a rough tape and caliber measures. So what are the conclusions from these sections, and what are the relevance for conservation? First of all, there are 3 distinct lineages among populations that were formerly considered a single taxon, Porteri on Santa Cruz. And it's important to consider that these 2 populations from Lacazette and Cerophata are distinct conservation units, and because they're distinct, it's important to warrant attention to the conservation status of this population in Cerophata, which is only 100 individuals. And this is not just a purely academic exercise, because if you don't name groups of individuals, populations that you think warrant immediate conservation attention, then the legislation you can't pass a series of legislative actions that will protect them. So you have to give them a name. And I think I put this picture in from a very I don't remember when. This is a Tuatara study, another reptile that's in need of conservation. And I think that this title is pretty telling. Bad taxonomy can kill. Naming organisms is important not only for an academic for academic purposes, but because otherwise, if you don't name them, you can protect them. So the other example, I wanted to shift gear again and just give you some example, revisit some of what Jeff said before about using DNA to help manage captive populations. And thanks to the fact that we have now a large database of genetic data from all the extant population for more than 3,000 individuals at this point, we can go around and try to identify the origin of captive tortoises with the hope to find precious individuals that can be used to supplement populations that are declining. So we are doing this with the unknown individuals that are kept in this pen from the Charles Darwin Foundation. We are doing this with the zoo animals. Some of these animals, remember that tortoises can live up to 150 years old, so some of the animals in captivity in the zoos have been there for quite a while and could be precious animals from populations that are species and that are threatened or with very small number of individuals remaining. So we have been doing that, and recently we have been revisiting the Lonesome George story. So Jeff left you with the idea that Lonesome George is indeed the last individual from its species on the island of Pinta. And he also mentioned that based only on mitochondria, remember at that time in 1999 we had only mitochondrial DNA sequences, and we knew that the closest relatives from Lonesome George were from the island of Hispaniola. And while Lonesome George is in the span with the females from the northern part of Isabella, from a population called Volcano Wolf, but it doesn't seem to be very attracted to them because it's not reproducing, has not been reproducing for the past 25 years. So we tried to study this a little bit more, and again, the museum's specimens were again crucial, and this time we had samples from the three skins from the California Academy of Science. We had samples also from four bone samples from tortoises that had been found in an expedition by Peter Pritchard in 2003 in a cave on Pinta. And so at this point we had a population sample from Pinta of six dead tortoises and Lonesome George. And at this time, we not only could extract information from the mitochondrial DNA, but also from the microsatellite. We were able to analyze the nuclear DNA. So we had basically, with 7 individuals, we could reconstruct the genotypic constitution in terms of frequencies of alleles of this extinct population. And we couldn't do that 5 or 6 years before, because we had only one mitochondrial DNA sequence from Lonesome George and 2 skins. The sequences were identical, so we didn't have frequency data. So with that, with this in our hands, we did some analysis, and I don't want you, this is too technical, and the takeout messages concentrate on this rectangle. This is the results of an analysis called, it's a structured bar plot, but basically what it says is, tells you the genetic composition of the individuals in each of 4 populations. So each bar that you see here is an individual. In this case, this blue bar means that this individual has 100% of his alleles belonging to the blue cluster. These individuals here in the red cluster means that all the individuals are assigned with 100% certainty to the population, to this population that's in this case Hispaniola. So here the genetic composition for 4 focal populations. One is Hispaniola here. These two are the 2 populations from Volcano Wolf. One is called PBR and one is called PBR. And this one is the composition of the Pinta population, the one made up of six skins and bones, 6 dead animals and 1 alive one. And one thing that you want to notice is that the genetic composition of the population from Hispaniola is pretty uniform. All the individuals are assigned to Hispaniola. They're all red. The composition of the individuals from the PBL population is also pretty homogeneous. Most of the individuals, the majority of the individuals, have all their alleles assigned to the PBL population. Pinta, there is a big solid block. All the 7 individuals are obviously assigned to this Pinta population. This population here, PBR, is the very intriguing one. It's a mess. A lot of individuals from PBR assigned to PBR, they're all blue, but some of them assigned to PBL because they are green, and some of them are hybrids, partly green and partly blue, meaning that there are individuals resulting from matings between individuals from PBR and PBR. But the thing that I want you to notice is that what we found in this PBR population, look at this guy, has lots of genes that are yellow and a little bit of blue. What does it mean? It means that these individuals that we collected on PBR, which is Volcano Wolf, actually has genes from where? Has genes assigned to the Pinta population, meaning that this individual is an hybrid. Is an hybrid between an individual from PBR and an individual from Pinta. So this individual is called PBR03 because it's our code, but this means that probably Lonesome George is not lonesome anymore. There is an individual, at least one individual, on Volcano Wolf that shares the same genes that Lonesome George had. The genes have the characteristic signature of the island of Pinta. So how might a Pinta tortoise arrive on Isabella? Well, Jeff told you already that we know that whalers and pirates occasionally moved animals among the islands, and we know that certainly this has happened very recently in the last 200 years. And the interesting thing, and I think it is very exciting for us, is that our sample of this population is one of the smallest one that we have. We have only 27 individuals from PBR. Why? Because it's really tough to go there. It's a very difficult volcano to climb, and when we went there, we didn't have enough water to stay for many days. So we stayed for 3 days, and we collected blood for only 27 animals. So, but on this island, on this volcano, there are more than 2,000 tortoises, and we didn't collect any of them. We just collected 27. However, out of 27 animals, we found an hybrid. So the chance there will be more individuals, and maybe even a pure Pinta specimen on that sample, it could be, it's relatively high. So what we're going to do now, next December, is to mount an expedition in which we're going to go over there and try to find more individuals, try to sample more individuals from volcano from this area and see where, if we find, first of all, this individual again, because we don't have it with us, and or more of the same type, or maybe even pure Pinta specimens. The next, but this PBR population, as I told you, is a very mixed population. It's also very mixed in terms of morphology. There are domed tortoises, saddlebacks, intermediate. So more recently, we have continued to work on the phylogeny and to try to reconstruct the phylogenetic history of this group. And what we have been doing with the help of this new postdoc that we have in our lab, that's a specialist on ancient DNA, we are trying to get DNA from all the extinct taxa. Jeff mentioned at the beginning there are 4 extinct taxa, one from the island of Floreana, Santa Fe, Rabida, and Fernandina. And what we found, and what I'm going to talk about is what we found about Floreana. So Floreana is a very interesting island. It's one of the southernmost islands and has a very long history of human colonization. There was a colony there of convicts since the 1800s. Has been used also, there was a little oil-producing industry in which they were killing tortoises to make oil for lamps, and so they depleted the population of Floreana, and very likely they brought to Floreana for oil production many individuals from different neighboring islands. The tortoises were extinct on Floreana, are extinct from Floreana. There are no tortoises anymore, and this is since when Darwin visited the islands, the tortoises were already gone. So what we did is to say, okay, where were the tortoises? The question we wanted to ask is, are the tortoises from Floreana, are they really a unique lineage, or the tortoises that we found on the slope, in the caves in this island, are they just animals that were brought from different parts, from other islands for the oil production? So we collected 25 museum specimens that were from 3 different expeditions. And what I wanted to point your attention is on this clade here, which include 18 of the 25 specimens of Floreana tortoises that we have. And as you can see, it's a single distinct clade. So this answered the first question that we have. Yes, Floreana, the Floreana tortoises was a unique lineage, a unique taxon, distinct from all the other taxon. So it is true, and the name, it's warranted that they have their own taxonomic rank. But the other thing that I want you to notice are these arrows. These are red arrows that are scattered throughout the phylogeny, are individuals that were collected in Floreana, but they clearly belong to different lineages. And these probably are individuals that were brought in from other islands, and whose bones were left in Floreana after they cooked the tortoises to extract the oil. So using DNA data, you can distinguish between the native Floreana stock and distinguish them from the one that were brought there from neighboring island. But the other I think most exciting finding is that you see this boxed, gray box, these are not individuals that are from Floriana. And these are not dead tortoises. These are alive tortoises. And where are they from? Well, they are clearly within the Floriana clade. So genetically, they are much closer to Floriana than to any other tortoises. But they were not collected in Floriana. They were collected in that same population on Volcano Wolf. So what this data shows is that the Floriana population is not, the Floriana taxon, call it a species or a subspecies, is not really extinct. Individuals from Floriana somehow, so maybe this human transportation was not so bad after all, at least for Floriana, because these animals from Floriana were brought over there and they mixed with the Isabella individuals, but their genes are still surviving. And so what I just wanted to conclude with my part saying that in this case, extinction may not be forever, because now what we can think is eventually to go back again on Volcano Wolf and try to find these Floriana individuals and start a selective breeding program in which by selectively breeding the tortoises we can rescue a species that was extinct for more than 150 years. And with this I will go back, I will leave this to Jeff.
But it's a successful captive breeding program. The tortoises on Espanola had almost gone extinct. They were bred in captivity, re-released on Espanola, and they're now reproducing. I just want to say that, and sorry to I have to skip over this. The final point, I do want to just say a couple words about the taxonomy or systematics of the group. As I said, it's a mess. Are they really different species? Well, it depends on what your concept of a species is. We know that they can hybridize. From a biological species concept, maybe you would consider them subspecies. On the other hand, we tried to emphasize the fact that they are good monophyletic groups from a genetic standpoint. So from so-called phylogenetic species concept, we probably should consider them different species, both for nuclear and mitochondrial DNA. Bill Gates maybe wants a giant tortoise named after him for a certain amount of money. We could do that. But we haven't done that yet. So as I say, I'm going to wrap up. Is there any economic value to giant Galapagos tortoises? And with that, I'm going to just recommend, leave this up as we take questions, a couple of very good books that summarize nicely both the history of work in the Galapagos on evolution. It's a very readable, nice book, and a recent book by Henry Nichols on Lonesome George himself as a conservation icon. And with that, I will end and be happy to take questions. I'm sure Gisela will be too.